relevant to this discussion is that HIV was found to be a retrovirus, something that was discovered only 10 years after the discovery of the polymerase. So now we can put together a life cycle for HIV. HIV as a free agent has reverse transcriptase inside of it. It has some other things inside of it that we'll come back to. It has on the outside of it little spikes, and those spikes recognize the surface of certain kinds of cells. We'll come back to which cells in a minute. And fuse with the surface of that cell, putting the inner components of the virus into the cell. Their reverse transcriptase gets activated, and it now makes a double-strand DNA copy of the RNA genetic information. That then, because there's an integrase protein in here also, that can integrate into the host chromosome, goes into the nucleus and integrates into the host chromosome. And that then encodes messenger RNA, because it's a DNA, can be read by DNA-dependent RNA polymerase, makes messenger RNA, and makes new genomic RNA. The messenger RNA encodes viral proteins. The cell gets viral proteins on its surface as well as inside it. You get budding out of the sort that I mentioned before, and you make new virus particles. That budding process is illustrated in this slide. Actually, this is not HIV, it's another virus, I think measles. But you can see very nicely how it buds off the surface, carrying with it a coat of protein underneath, or actually going through the membrane, carries the genetic material inside it, and ultimately it'll pinch off here and release a free virus particle. Now, with HIV, the virus particle has a very strange property, and that is that this, what we call the nucleoid inside the particle, is not symmetric. In almost all of the viruses, this is spherically symmetric. Here, it's this asymmetric structure for reasons that are not clear, but represent a signature of HIV. So this is a nice diagram of the HIV particle. You can see the spikes on the outside. This is what attaches to cells. You can see the genome, which is actually 2 molecules of RNA. You can see the reverse transcriptase in here. You can see the integrase, which is going to help the double-strand DNA go into the chromosome. And you can see another enzyme, a protease, that's important for maturing the virus. Now, you can't talk about HIV without talking about how awful the HIV epidemic and the AIDS epidemic has been. 65 million people have been infected at this point. The data is actually 2005 data. 25 million people dead, 40 million people living with AIDS, 5 million of those in India and about another million in China, as far as we know. And I call those out because those are the most populous countries in the world, and they are spreading the virus. Today, there are about 13,000 infections per day, mostly still in Africa, 5 million a year. 3+ million people dying every year of the viral infection. That's as much as the 2 other infections that have been a scourge for many years of the less developed world, tuberculosis and malaria. The effect on African countries has been dramatic and awful. In some countries, seeing a reduction in average lifespan of as much as 20 or more years. So why is this so lethal? Fundamentally, it is a classic non-equilibrium virus. Actually, HIV, or a very close relative of HIV, almost certainly its parental virus, is endemic in Africa in great apes, found in chimpanzees. It got a foothold in the human population some significant length of time ago. Some people say as much as 70 years ago. Probably was spread at very low levels, maybe very inefficiently, and not recognized because it doesn't cause a characteristic disease. It causes things that look like a lot of other things. And then came to the United States, probably on an airplane, around in the 1970s, and started killing people in numbers that were large enough that they were recognized by physicians in the late 1970s. So why is HIV lethal? Well, it grows in one of the key cell types of our immune system. So our immune system has lots of different cells in it, but one of them is a helper cell that helps everything else take place. And if you don't have helper cells, the immune system is pretty puny. And HIV has targeted itself on helper cells. So infected people lose their helper T cells over years, because it takes that long for the virus to get through the cells, and ultimately they become immunodeficient and they die from infections that are caused by organisms that healthy people would ordinarily just get rid of. And so I've taken us from a very general consideration of viruses to the very specific virus HIV, virus that grows in helper T cells and causes lethality in humans, a virus that we have to stop. And yet we have found it very difficult to stop its transmission. Why is that true? That will be the subject of the discussion in our next two parts. Thank you.